In this video we will discuss about N-glycan biosynthesis process. In some of our previous videos we have discussed only about this endoplasmic reticulum, how the glycan N-glycan post-translation modification begins in the endoplasmic reticulum, and then it further goes to Golgi for further post-translation modification or further modification of N-glycosylation. In this video we will discuss both the post-translation modification in endoplasmic reticulum and Golgi in terms of N-glycan. So what happens in endoplasmic reticulum is that this glycan, preformed glycan, that is, some part of the glycan is formed outside the ER lumen and some part is formed within the ER lumen, and this glycan will bind to the the co-translation, bind to the protein that is translated, that is either in the process of translation or is recently been translated, and it is bind to a specific site of the sequence of amino acid, that is asparagine, then any amino acid, then serine or thiourine, and this any amino acid should not be proline. So in that condition, this glycan will bind to that site asparagine of that sequence with the help of OST. Now after that, since this is trimethylated glycan, the alpha glucosidase 1 and alpha glucosidase 2 will trim off one glycan each, and this monoglucosylated glycan will then undergo structure modification in the presence of calnexin, calreticulum, protein disulfide isomerase family such as ERB57 to form a native structure and have a native disulfide bond. After that, this glucosidase 2 will trim off another mannose that is present in the glycan structure, and after being verified to have a structure close to the native structure, the protein will then move forward to the Golgi for further post-translation modification with the help of this COP2 transport vesicles. And this COP2 transport vesicles move the protein into the cis part of the Golgi, where further trimming of this mannose takes place with the help of alpha mannose 1 and alpha mannosides 1, the help of alpha mannosides 1, and this GNT1 and GNT1 then results in addition of this glycan to the glycan structure, and this alpha mannosides 2 will trim off further mannose in this cis Golgi region. After that, it will move into the medial Golgi region, and where with the help of GNT2, there are two conditions that are occur. First, with the help of GNT2, another addition of this glycan occurs, and after that different kinds of modification occurs. First is that with the help of GNT4 and GNT results in additional glycan and then galactose, or with the help of GNT, the addition of only galactose, and GNT3 will leads to addition of additional glycan in between, and that results in afterwards the GalT will leads to addition of galactose. So all this will be the precursor for the complex N-glycan, whereas one condition is that after the GNT1 in the cis Golgi region, it can directly go to the medial Golgi region. That results in the addition of galactose to this glycan, and this further when it moves the trans region, this leads to the addition of sialic acid. This can be alpha 2,3 sialic acid or alpha 2,6 sialic acid. This leads to the formation of hybrid structure, and in other three cases, what happens, the addition of sialic acid will leads to the formation of complex structure, as it has all the available monosaccharides, but not in a defined structure, as all of these have different kinds of pattern. And in this case, the amount of different monosaccharides are varied significantly, such as sialic acid are only one, whereas mannose are quite high. So this leads to the this structure is called hybrid and glycan structure, where the carbohydrate or sugar structure is in the form of hybrid. So, or we can say the amount of monosaccharides varied significantly in hybrid structure compared to the complex and glycan. That's why it is different from the complex structure. And there could be other reason also that can be understood from the reference that has been provided in the description. So that's all for this video. Thank you for your time.